everybody, we're going to go over our DNA notes today. Make sure you're using in your science notebook the 3 slides that you have. There's some parts where you have to type, and then there's some drag and drops for the pictures for you. All right, so DNA stands for deoxyribonucleic acid. It's one of the 4 types of macromolecules that we discussed last year in class. So DNA is quite plentiful in your body. You have about 2 meters of DNA in one single cell. And if you remember, cells are microscopic. We can't see them, but you have about 6 feet, 6 inches-ish worth of DNA inside that cell, inside the nucleus. And you guys, on average, have about 75 trillion cells. That means I could get your DNA, if I strung it out from end to end, from the Earth to the sun, more than 400 times. So even though if we strung it out, it's super duper long, it would be very, 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 very thin. So you can see the diameter down there at 0.00000002 meters. So really thin. So the question is, well, then how the heck does all that DNA get in there? So if you have about 6 feet of DNA in length inside a cell, and your cell is microscopic, how does that work? Well, it does help, again, that it is super duper thin. But what it's doing is winding itself up. So it's kind of like a ball of yarn or floss. If you've ever unwound a whole ball of yarn, it's really, 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 really long. It could be yards or meters long. But when you wind it all up into a ball, it can take up this much smaller space. And that's kind of what DNA is doing. It's winding itself around. There are helper proteins in the cell that are helping it do this. So you can see in the picture here that you've got the DNA in blue winding around these helper proteins. So again, DNA is inside our nuclei. So you can see in the 2 microscope pictures on the bottom, the dark purple areas are the nuclei. In organisms that don't have a nucleus, so prokaryotes, the DNA just kind of floats around in the cytoplasm. So the benefit to having a nucleus is just kind of an extra layer of protection. The downfall energy to maintain that part of your cell. So there's trade-offs to having a nucleus and not having a nucleus. And so for bacteria, the trade-off of having a nucleus is not good enough to outweigh what they're doing. So DNA under an electron microscope, you can see this picture is from an electron microscope. That is what it looks like. Kind of just looks like thin, stringy stuff. When we did the DNA extraction, it kind of all clumped together because there were different things that we did to the DNA to help kind of pull it out of the nuclei and then make it clump together so we could actually see it. You can't see DNA otherwise if you just have like a string of it. It's too small, too thin. And again, super tall. All right, so what's its actual structure? So DNA is pretty cool in that we only discovered it in the late 1800s. So a scientist in the late 1800s, 1890s, I believe, discovered it and didn't think anything super remarkable about it. He called it nuclein, I believe, because he knew it came from the nucleus of the cells. So he was like, okay, great. It wasn't until the 1920s that scientists started to figure out that DNA was actually the molecule that determined all your traits and who you were like. For a long time, scientists were like, this is just kind of a super simple molecule. How could this possibly have enough information in it to determine all the different traits we see in humans? And not only humans, but like in plants and your cats and your dogs and the bacteria that are growing inside you. So once they figured out that DNA, in fact, did appear to be what was the molecule that had all that information, they then really wanted to know, okay, we need to know exactly what this molecule is like. And so they discovered in the 1920s a little bit more about its structure, but they still didn't have a clear visual of what this thing would look like. And so we entered the late 1940s and early 1950s with these 4 scientists. So we got Rosalind Franklin here, Watson, Crick, and Maurice Wilkins. So they were going about discovering DNA in different ways. Rosalind Franklin and Maurice Wilkins were working on X-ray images of DNA, whereas Watson and Crick were working more on 3D modeling. Watson and Crick did use Franklin's X-rays, so you can see there's her famous X-ray up in the corner, without her permission to help guide their model, and unfortunately did not credit her work when they published their results of what DNA ended up looking like. Also, even perhaps worse, they did not award Rosalind Franklin the Nobel Prize, the Nobel Committee, when they awarded it in 1963 maybe, to Watson, Crick, and Wilkins. And the reason she was not recognized was because she had already passed away, and they don't award the Nobel Prize to people who've passed away. But pretty sketchy. They have since credited how important her work was, but during her lifetime they did not. All right, so there's our people. Okay, so DNA is a polymer. So remember, a polymer are super, super long chemicals made up of repeating parts or repeating atoms. So we looked again at polymers last year. Some of these examples we looked at included proteins, but we also looked at, in my class at least, we did diapers because they have a polymer inside them, silly putty, things like that. So the key part to a polymer is you're going to see the repeating parts. So they're going to have parts that repeat over and over and over and over again. All right, so the shape of the molecule, just in general, when you look at it from afar, it looks like a twisted ladder or a twisted zipper. We call this shape the double helix or a double helix. You've probably seen that shape all over. It's pretty iconic. So the parts that DNA is made up of are called nucleotides. So these are going to be the parts that are repeating over and over and over again in the DNA molecule. All right, so a nucleotide is made up of different parts. So there's 3 parts in the nucleotide. So the first part is the sugar here. So the sugar is in blue, the phosphate here is in orange, and the base is in green. So these are actually made up of atoms. So this, for example, sugar has a carbon here, carbon here, carbon here, carbon here, carbon here, with some hydrogen sticking off of it. The phosphate has, for example, phosphorus atom with some oxygen sticking off of it. And the bases are primarily carbon, hydrogen, and nitrogen, as well as some oxygen in there. But the thing is it looks really weird to draw the atoms. So if you guys remember last year, we kind of drew like the molecules. You would draw like carbon attached with a line to carbon. We can do that, and you'll see on later slides examples of that. But it gets really complex to draw those molecules, so we tend to use shapes to make our lives easy. So this shape, the pentagon, is always going to represent the sugar. It is a 5-sided sugar. That's why we use it, pentagon. And then we always use a circle for the phosphate. The base itself, you'll see a variety of shapes for that. Okay, so this nucleotide, this is the thing that's going to repeat over and over and over again. So if we look at that ladder, you're going to see on the side the phosphates and the sugars. So they're in yellow and green here. So they're just repeating phosphate, sugar, and then there's the base, phosphate, sugar, base. Remember, one of those is a nucleotide. The DNA molecule could have hundreds of thousands of these, okay, or millions even. So we got sugar, phosphate, base, sugar, phosphate, base, sugar, phosphate, base. Same thing's happening on the other side. The bases stick off the sugar. They're always off the sugar. They stick off, and they're chemically attracted to each other. So their atoms want to join, so they form bonds between them. Okay, these dashed lines represent bonds between the bases, and that's what keeps the ladder together. Okay, so we've got sugar, phosphate, base joined across to a base here, a sugar, and a phosphate. So there's only 4 different types of bases. So we've got 4 bases called adenine, guanine, cytosine, and thymine. We call them A, G, C, and T. You are welcome to use those abbreviations pretty much whenever. You'll notice in this drawing here, the sugar and the phosphate are shown as just a line. We don't even draw the circles and the pentagons anymore because they never change. So why should we draw them out all over and over and over again? Instead, we just draw it as a line, okay? And then the bases are joining in the middle. Again, the bases are not joined physically, they are chemically bonded. So they're bonded through chemical bonds that we talked about last year. So if we go back real quick, you'll notice there's different shapes on these bases. So we usually use different shapes to represent the 4 different bases. So we've got, like, let's say that's base A, that could be base T there, that maybe is base C down there. So you'll see different shapes to represent the different bases. Again, this is the only difference in the nucleotides. The sugar is always the same, the phosphate is always the same, and there's always a base, and it's always one of these 4. All right, so again, here is a nucleotide with the sugar in the pentagon shape, the phosphate in the circle, and the bases in the different shapes. What's really interesting is because of the way the atoms work, the bases will actually only bond with one other base. So adenine and thymine will always bond together, and cytosine and guanine were going to bond together. So this was discovered by the scientist sitting off to the right there. That's Erwin Chargaff. So you'll notice if we go back up to this slide, you'll notice this drawing has them, so they're complementary. So you can know that, okay, C is going to pair into G there, A and T are going to pair. And if we go back up one more, you'll see G and C together, G and C together. There's a T and an A, there's an A and a T. These are the bases pairing rules. You'll always see, again, G with C and A with T. And it just has to do with the atoms and the way the atoms are able to bond with each other. All right, so here's kind of what a diagram would look like. So you can see one nucleotide, phosphate, sugar, base. Here's another nucleotide, phosphate, sugar, base. Here's a third, phosphate, sugar, base. Same thing's happening on the other side of the ladder, phosphate, sugar, base. And then the bases have a bond between them to keep the ladder together, okay? Again, our base pairing rules are T's go with A's and G's go with C's. Here's what the bases look like in terms of their atoms. You can see why we just use generic shapes, because that would get kind of annoying to draw. Also, anytime you just see, like right here where the 2 lines just kind of intersect, there's actually a carbon atom that we don't even draw. And then sometimes there's hydrogens too that we don't indicate with our drawings either. So because these are so difficult to draw in terms of their atoms, we just use colors or shapes to represent them. So in this case, again, they've used purple and yellow for adenine and thymine, so you can see that they complement each other with their shape because they bond together. All right, so I'm going to have you just real quick on scratch paper somewhere, see if you can match this sequence based on the base pairing rules that we have. And if you are confused, I'm not asking you to say, like, you know, this letter will find a base here. I'm saying what would be across from it? So like right here, what would be across? What would be over here on this side? All right, so here's the sequence. So going down, T would be across from that A, then an A, a G, a G, a C, a C, a G, an A, a C, a T, and an A. That's what would be across from each pair in the ladder. All right, so these next few slides, you have very specific stuff on your notebook page to make sure you're filling out with these. All right, so DNA is often called the blueprint of life. So in simple terms, you're going to make sure you understand that DNA has instructions for how to make things, specifically proteins. And if you remember from last year, proteins do a ton of different things in the body. They do everything from like generating skin color and hair color and eye color to creating some of the chemicals that your brain uses to communicate, to creating the things that your digestive system is using to break down your food. All those instructions are going to be in the DNA for how to make those proteins. So the only thing in the DNA that is changing ever are those bases. And so scientists pretty much figured out, oh, well, that's where the code has to be then. Because if, for example, I have blue eyes and you have brown eyes, well, then we obviously have different codes or different recipes for that, right? So where could that code be? Well, what's changing in the DNA? The bases. And so it turns out that if we look at the bases, we see differences in people for who have brown eyes and blue eyes or green eyes. You see the bases are going to be different from each other. All right, so again, that's just kind of reiterating, we need to look at the bases. That is where the code for making your proteins is. So the area of DNA that will code for a protein is called a gene. So a lot of people have heard this word before and don't actually really know what it is. Most people think all of DNA has codes, and that's not true. There's portions of your DNA that don't have directions for how to make things. It's called junk DNA. It's not actually junk. It does things, but that's more for high school. But sections of DNA that have codes in them, like if you look at your DNA and you're like, okay, this section right here has my code for how to make stomach acid, let's say. That is called a gene. So genes are where the codes in the DNA are. The bases we look at, again, will code for proteins. And we're going to go over how this happens soon. And then the proteins can help determine your traits. So the protein that you're making will determine, your eye color, your hair color, or things like that.